Hi, I'm Christine, and today myself and the members of the JUNO and GPS2 projects based at the Wellcome Sanger Institute will be presenting to you tips for interpreting phylogenetic trees as part of the free online training offered from these two projects. Great, and I'll just hand it over to Steve to give you an introduction of both the projects. Thanks.
Thank you, Christine. So Christine's given me 10 minutes to give you some background on our project and what brings us all here to these courses and this webinar. So the story begins quite a long time ago. So back in sort of 2009, 2010, 2011, and that was the period when we got the GPS project going. So some of you on this call will be involved in GPS, some involved in the JUNO project, and some involved in both. And I think maybe a few, maybe even involved in neither so far, but hopefully we can bring you in. So GPS, that's the Global Pneumococcal Sequencing Project. That dates back to started in 2011 and was really motivated by the idea of wanting to prove the principle that genomics could be useful in surveillance of a pathogen, and to do that on a global scale. So Pneumo is obviously an important global pathogen, so it turned out to be a really good place to start. So it's an important global pathogen in terms of burden of disease. But then the other interesting feature that made it ideal for what we were wanting to do was the introduction of vaccine around the world around that time. So essentially what we were aiming to do was to say, okay, can genomics be useful in helping us to understand the response of the pathogen to the introduction of the vaccine in multiple different countries. And you know, you don't propose to do these kinds of things without having some kind of confidence in what the outcome might be. And we were fairly confident from work that we'd already started to do and projects that we'd already run. So we knew from one project that if we look at a collection of one strain of pneumo, we could use genomics to really drill down on recombination events and things that were happening in terms of the evolution in response to antibiotics and also vaccines. So that was looking in one strain, and then we looked with colleagues in Massachusetts at a species-wide perspective to say, when we introduce the vaccine, what do all of the strains collectively do? So we'd done that that preliminary work, and then along with Keith Klugman was the project leader at the time, we went to the Gates Foundation, and the Gates Foundation were sold on the idea. So that was when the initial funding began. And as I say, the aims at that time were really to just prove the principle that this can be useful. And then, so some years later on, we've been able to produce some high-profile papers that really say yes, that this works, that genomic data is useful. And over that period of time, we've seen lots of other similar projects on lots of other different pathogens. So this is now widely accepted in clinical microbiology, I guess, that genomics is a powerful tool in understanding the evolution of the pathogen in response to clinical interventions and identifying high-risk strains. So that first phase of GPS was a success. We were funded to produce 20,000 genomes. We produced in the end more like 27,000 genomes, and we cover more than 50 countries across the world. So, you know, tick, I think we did a good job there. Then the next phase that we've recently begun, that funding started at the beginning of this year for 4 years. That is to shift the focus and say, okay, we've proved the principle, we know this can work, but the way it worked for GPS for the first phase was all of the sequencing went on at Sanger. And to make a sustainable system going forwards, you need to decentralize that sequencing activity. And that has motivated a lot of thinking about how do we not just get sequence data generated in many countries around the world, but also how do we get the data analyzed? So that's motivated a lot of the thinking around training and a lot of the thinking around the material that Christine has been important in putting together to help with that training. So the online stuff, etc. So I've not mentioned JUNO so far, but JUNO was something that arose alongside the GPS project because I guess really because one of the important things was a strong interest from the Gates Foundation in the program that was run by Keith Klugman. That program it's the pneumonia program, but if you read their website, they state that there's an interest in three important things are pneumonia, meningitis, and neonatal sepsis. So neonatal sepsis, their interest in neonatal sepsis meant that Group B strep goes high on the agenda. At Sanger, we had in the past run a few genomic studies on Group B strep, Strep A galactii. From a personal point of view, I'd always been really frustrated that we hadn't been able to really pursue those because A galactii is one of those species that gets less interest. So it's harder to persuade people that we should be spending our research budget on studying that pathogen. So I was delighted when I realised that the Gates Foundation were also interested in Group B strep, and we had a conversation and realised that actually we could prove that same principle that we did with Pneumo and use genomics to do a survey of Group B strep, a global survey of Group B strep. And that was really interesting because we're at a different stage then. So the vaccines are likely to come along for Group B strep and are in development, but we've kind of got ahead of the game a little bit with Group B strep because we can actually do that global survey and then try and predict how the pathogen will respond to the vaccines that will be introduced. And the timing works really nicely because to be able to say we'll do a global survey of a pathogen and predict how it's going to respond to a vaccine, we wouldn't have been able to confidently do that if we hadn't had the experience that we've gone through with Pneumo. So Pneumo is like training for other pathogens, and now with Group B strep, we can apply that thinking to being able to forecast what's going to happen from the genomics. So that's where we are. And again, so for JUNO, there's also a strong focus on training. So both of those projects have generated or created large networks of partners around the world, which is one of that alone makes me really happy and proud of what we've achieved through these projects. And I think we have a really good relationship with our partners, and these training activities are being really well received. And I think they're going to really make an important change in the way genomics is done on a global scale. As I said before, we don't want to remain in a situation where all of the samples come to one central location in a rich country and everybody has to rely on that resource. We want the sequencing and the analysis to be done locally, and that's what these courses are aiming to achieve. So I think, Christine, I hope that's the kind of introduction that you were hoping that I could give. I really enjoy talking about this kind of thing, and I can go on for a long time, but in the interests of the webinar itself, I will stop there.
That was fantastic, Steve. Actually, that was exactly what we wanted. If there are any questions, again, please feel free to add them in the chat box. But are there any specific questions to Steve? Feel free to unmute yourself. The room is fairly quiet, but we're a very relaxed bunch and friendly bunch. So feel free to just kind of ask questions as you go and in the chat box. And again, please fill in your country and where you're from. We'd love to know the network and that we're trying to build with both these projects as Steve has introduced. Without further ado, I'd like to introduce Steph, and she'll sort of jump in and talk about how to interpret trees for specifically strep pneumo. There we go. Over to you, Steph.
Oh, thanks, Christine. May I share the screen?
Absolutely.
Can you see my screen now? Yeah, thank you. So here are some tips for creating and interpreting a pneumophylogeny. So first thing first, no matter you are constructing or reading a tree, you should always bear in mind about QC. And there are two questions you could ask. Is the sequence data pneumo, or is there any contamination? And contamination could be caused by any other species. And it also could be due to a mix of more than one pneumococcal strains in a single sample. And here are two examples. So in this tree, you can see the yellow samples that have very long branches. And the QCs that shows that these samples have only 25% matches with pneumo reference sequence. Therefore, they are very unlikely to be pneumo and should be QC failed. In the other example, there are some green samples like this that do not have branches after their internal node, which are very strange. And this sample had a very high heterozygous SNPs, which strongly indicating that these samples are a mix of more than one strains in a single sample. So if you are constructing a tree, remember to do QC first. There are two types of trees, species-wide tree and lineage-specific tree. In this tree, in this species-wide tree, it includes genetic diversity across the whole bacterial collection. And this tree includes multiple GPSCs and clonal complexes. For example, if I have 500 samples from a country, pre and post PCV vaccine, then I create a tree to show its genetic diversity. And this country tree is a species-wide tree. And in this country tree, GPSC8 and clonal complex 289 is a predominant strain. However, in a species-wide tree, it is a bit difficult to see the variation between isolates that belong to one GPSC. The resolution is not high enough to tell the difference. In that case, we can construct a lineage-specific tree. And in this tree, the isolates are all belong to one single GPSC. And if you are interested in seeing more examples, you can visit the GPS website. And in the next slide, let's see why lineage-specific tree has a higher resolution. So trees are built based on SNP patterns, and each cross here indicates a SNP. There are 3 samples in each tree type. Remember, species-wide tree has high genetic diversity. There are 3 samples, represent 3 lineages in the species-wide tree. These gray color reads are sequence reads shared among all pneumos, or we can call them core reads. Lineage-specific reads are in other colors. For example, purple reads, green, blue, or orange. To construct a tree, we first mapped our reads to the reference and then extract SNPs. In a species-wide tree, a generic reference is used. Sample 3 map very well to the reference because it is similar to the reference. However, sample 1 and sample 2, which do not similar to the reference, only the core reads in gray can map to the reference because their purple and blue color reads are very different from the reference. And then the alignment tool will fill the gaps with ends, and no SNP will be extracted from these regions. In that case, only SNP in the core genome were extracted. In contrast, the lineage-specific tree, samples are similar with each other and a lineage-specific reference is used. Therefore, more reads can be mapped to the reference, and more SNP can be extracted to build a tree with more genetic variations information. That's why the lineage-specific tree has a higher resolution. Root is important for a tree, and so does it for the phylogenetic tree. So rooting phylogeny at an appropriate position is critical for interpreting a tree. There are two methods. The first one is midpoint rooted. This is used when we don't know any outgroup. This is not the best way because it assumes all of your samples are evolving at the same rate. The root is rooted in the midpoint between the two long branches. And a better way to root the tree is using an outgroup. To do so, you can include one or more samples that you know they are known to be outgroup. In Pneumo, classical non-encapsulated pneumococcus is usually the choice. And here is the figures to illustrate why. And these figures show the pairwise comparison around 600 Pneumo in terms of core genome and accessory genome. And genomes that are more similar to each other has a smaller distance. Each point is a comparison of two genomes. So there are many points, but they are grouped in different colors. So let's look at the red points first. The red point close to the origin, they were highly similar to each other in terms of the core and accessory genome, and they form a monophyletic cluster in the tree, or we can say they are from one lineage. The blue points here show the pairwise comparison of isolates belong to different clusters or different lineage. Therefore, you can see that they have a longer distance between each other. Last but not least, the green dots show comparison between classical non-encapsulated Pneumo and the others. They are clearly a divergent from other Pneumo, and the discontinuity between the green dots and the others indicate that this non-encapsulated pneumococcus has distinct signature in their core genome and also in their accessory genome. That makes them a perfect outgroup in the tree. Another thing to bear in mind when choosing outgroup is not to choose something really distantly related to your example, for your samples. And for example, if your genomes are carrots, you may want to choose parsnip as outgroup, but not banana. It is because two different would mean there are hardly any roots mapped to your reference genome. Now you can root your tree on MicroReact by right-click in the tree panel. You can do midpoint rooted, or you can choose the outgroup and make it as the root of your tree. The last tips for Pneumo tree is that the whole genome clustering method, GPS2, MLST, and phylogeny, they are usually in concordance, like this country tree. But there are some rare examples we observed in the GPS dataset, but we are talking about looking at 13,000 genomes in the GPS dataset. We've found some rare examples. Some isolates belong to same clonal complex, could be distantly related in a phylogeny, like this. They are in different location of the tree, but they belong to the same clonal complex. And this could be due to the fact that the 7 housekeeping genes chosen in the MLST scheme, they are also frequently undergoing recombination. But the phylogeny usually in concordance with GPS-C, which take both core and accessory genome distance into account instead of just only 7 genes. So this is the end for the Pneumo and phylogenetic tree, and I will hand it over to Christine. Thank you.
Thank you so much for Steph. There is a wonderful, one of the reactions is a clapping emoji. So yeah, please do clap for our participants if you don't want to unmute. Yeah, woo. I always like that, even though you can't hear the clap, the sentiment is felt. Thank you so much for that, Steph. Okay, you can save your questions till the end. So I'll move on to Dorota, who will be telling you more about phylogenetic trees and interpretation specifically for strep A galactii. Thanks. Over to you, Dorota.
Yes, thanks, Christine. Just to let you know, I'm not gonna go into presentation mode because I find that my Zoom breaks if I do that. So I'm just gonna have it as slides if you guys don't mind. So I just wanted to start by saying that sort of Steph's overview into phylogenetic analysis in strep pneumo. It's a lot of the tips are universal, so I would say they definitely apply for group B strep and also other pathogens. So things like doing QC analysis, advice on routing the tree, and the different differences between sort of a species-wide tree and a lineage-specific tree, that all applies. So I'm not going to cover that in this on my slides. I would just focus on two things in terms of GBS phylogeny. So phylogeny of all GBS isolates in the data set, or a species-wide phylogeny, and then also a few pointers about phylogeny of individual GBS lineages. And again, some of the things that I'm going to mention, I think they are also universal, so they're not necessarily just GBS specific, but you can also use that those tips for other species. So in terms of doing phylogeny of all GBS isolates in a data set, a few things that I would sort of point out as tips is that, again, watch out for contamination. So like Steph mentioned, presence of very long branches, or maybe nodes that don't have any branches whatsoever. I find whole species phylogeny very useful to evaluate clonal complexes, basically how those isolates are distributed on a phylogeny. And how the clustering of different softwares looks if you compare with the phylogeny. And I would say to be very careful when you use a whole species phylogeny if you want to evaluate phylogenetic relationship between different GBS lineages. So just to give you examples of what I mean by this, starting by spotting contamination in the data set. I would just like to say that doing QC is probably best to do it at a very early stage of, sort of reviewing your data set even before you go into phylogeny, just so you don't waste time and resources building a phylogeny that contains some contamination. But if some samples, if some samples sort of past that your initial QC analysis, but they still look odd on the phylogeny, then it's sort of a red flag to maybe go back and revisit your QC steps. So this is a phylogeny of around 700 GBS isolates from maternal carriage in the UK. So I was a bit lazy here. It's midpoint rooted rather than rooted on an outgroup. And the tips of the branches are marked with whether the samples have failed or passed QC. So you can see sort of at the bottom of the tree, there are some very long, unusual branches that, those were actually a contamination. It was either a wrong species or a group B strep contaminated with another species. But just to say that if you do see something like this on the phylogeny, a very odd branch, I mean, there might be a biological explanation for this, or might be like a hypermutator. So I would just use it as a red flag and then go back to your data and evaluate your assemblies. Are genomes unusually long, or is there any evidence of contamination in the data set? Moving on to the next point, which is using a whole species phylogeny to evaluate clustering and grouping. So this is what I found whole species phylogeny is particularly useful for. So again, this is a midpoint rooted tree of my collection of group B strep from maternal carriage, but this is now a revised data set. So I removed all the contaminations, and it looks much neater. I don't have those long branches, and you see a better resolution of individual clades. So the phylogenetic tree is annotated with clonal complex assignment based on MLST in the first column, and then the second column is results of pop-punk clustering I think pop-punk is something we're gonna cover in later training. So I'm just mentioning it here, but you know, don't get too worried about the fact that you don't really know what pop-punk means, because I think we'll be covering in training later on. This is just to show that, you know, if you put sort of this information on the tree, you can see, for instance, what colon complex is. That okay, all my isolates that are, for instance, belong to colon complex 1 based on MLST, those isolates actually cluster very nicely on the phylogenetic tree together. So it gives you sort of confidence that that clustering you know, worked well, and the from phylogenetic perspective, isolates are also closely related. In terms of pop-punk, there is some variation there, but you know, it's not necessarily a red flag. It just means that pop-punk detects some sort of heterogeneity within that group that MLST doesn't, and that's just basically the nature of MLST analysis versus pop-punk analysis. And while I'm on this slide, I would like to change here and my display and show you something in MicroReact, which I found very useful. And I think this is sort of a new feature of MicroReact that I actually just discovered it preparing those slides. So I'm moving now to MicroReact display. This is the same phylogenetic tree that I showed on my slide. But what you can do, so this is something that is very useful for a quick visual overview of how your isolates cluster in the phylogeny versus your MLST or pop-punk clustering. So you can see in the right, I'm gonna move it slightly, in the right-hand corner, you have a legend of what the different colors means for the different bars. Now, if I click on the internal nodes of some of those clades, so I'm gonna go from the top. So I'm clicking here on colon complex 1. And what happens in MicroReact is that all this bottom information disappears, and it only displays the information for the node that I clicked on. And it gives you a nice visual overview. It shows that these are all colon complex 1, and then it displays the pop-punk assignment for the isolates within that clade. And you can do it for all the other nodes, which again, I found really interesting and useful for quick visual overview of how your isolates cluster on the phylogenetic tree. So just going back to my slides now. So moving on to the next point, which is using whole species phylogenetic tree to evaluate phylogenetic relationship between different lineages. So I would just want to say, please be careful when you do that, because the topology of the tree that you have will really depend on the method that this has been used for the reconstruction of that tree. So these are two different trees that are actually based on the same data set and the same list of the same alignment and the same composition of SNPs. But I use two different methods to reconstruct that tree. I'm not gonna go into the details of what those methods are because it's not really relevant here. Just wanted to say that if you look at the bottom of the tree, so you have two colon complexes, colon complex 23 and colon complex 17. And if you look at method 1, it looks like the phylogenetic, those two clades sort of split from an ancestral node into sort of two sister clades, whereas using method 2, it looks like actually CC17 emerged from CC23, and that CC23 is sort of ancestral background to CC17. Now, I'm not gonna sort of speculate which is right or wrong, because I think it requires a little bit more analysis, in-depth analysis, comparison of different models for phylogenetic reconstruction, but just to sort of highlight that, you know, depending on the method you use, you might get different answers. So please be careful if you want to use that sort of if you want to use that to make some sort of conclusions about sort of relationship between different lineages. And finally, looking at the phylogeny of individual group B strep lineages. So there's not really much to say other than that different GBS lineages show different level of genetic diversity. So for instance, those of you who work with group B strep will be familiar with lineages such as colon complex 17 or colon complex 1. So CC17 tends to have relatively low genetic variation in comparison to other lineages such as CC1 or CC19. And again, without going into too much details, just to say that this is associated with a varying level of recombination between the different lineages. So when you do a phylogeny for those individual lineages, expect to see slightly different topologies for those phylogenetic trees. So the way you interpret those phylogenies will really depend on the lineage that you're working with. So there's sort of not one size fits all. So for instance, just to show here some examples of 3 different lineages, we have clonal complex 1, in the middle clonal complex 19, and then clonal complex 17. So you can see that there are slightly different sort of topologies of the trees, which CC17 having sort of 2 main dominant clades in this sort of comb-like structure, whereas with CC19 there's a lot more genetic diversity with sort of tight clusters and also more lineages on the phylogeny. So more sublineages or subclades within the phylogeny. So just something to bear in mind that depending on the lineage you work with, you will see slightly different tree patterns. So yeah, how you interpret one tree might be different to how you interpret the next tree, because again, it depends on the level of genetic diversity you have within that lineage. Yes, and I think that's all from me in terms of Group B strep. Happy to take questions if there are any.
Does anyone have any questions for Dorota? I'm going to jump in with one. So Dorota, on slide number 5, sorry, you might have to share your screen again.
Sorry, I killed it too quick.
I was too keen there to exit stage
to move on.
Maybe you, on question, slide number 5. Maybe you answered this whilst I was doing some stuff. So let's say I run a phylogenetic analysis and I do method 1 and I don't think to do method 2. How would I know which one is correct? Is there like a simple answer, or it becomes very complicated?
Yeah, so I was worried that if I do this slide, it's all open a Pandora's box of not being able to follow up on it. And I'll be honest with you, I haven't actually done analysis like that myself, but I'm mentioning this because I have seen papers where, you know, there were some conclusions made about relationship between different lineages, and I think you have to be careful. I mean, so if I was to sort of rely, say, one method over the other, I would say method 1, because I used an algorithm. I used IQ3, which actually has the software itself selects the best model for reconstruction of the phylogeny. So it sort of it does the model selection for you. But if you select the model yourself, then you have to really be able to justify why you selected that model. And I think it's just a case of comparing lots of different models and see what answer you come up with. But I don't feel particularly strongly about one method telling you, okay, saying that this is the best method to use. I think it really depends on a sort of level of diversity within your tree. And I would say just compare different methods. But yeah, it's not really answering your question, is it?
No, it answers it perfectly. And we'll just link to that in the subsequent modules, we'll be using IQ3, which will select the model base for you. And also the models are fairly mathematical, unless you've studied math, they're quite complicated. So I would always choose the program that chooses the method for you, because it's justified within the program itself. And another important thing that you've raised, the phylogenetic tree is hypothesis generating. It is not a be all and end all. That's, I think, one factor that we always have to remember. You're supposed to generate a hypothesis, so it's not like rule. It gives you an idea of what's happening phylogenetically or relationship, but it's not the answer to everything. Do you guys agree with that?
Yeah, I think I think that's a very good point. So like if we were to rely on a phylogeny from method 2, then the hypothesis might be that CC17 emerged from CC23, but I would say, okay, can I have more evidence to prove that, to demonstrate that?
What would be the evidence? What would you want to throw in there? Would you want to throw older samples or something like that?
I would probably look at, maybe recombination analysis, see if there's sort of movement of genes from CC23 into 17. But I don't want to go into more details, because I haven't actually done analysis like that myself. So I will be just speculating on what could be the next step.
No, thank you so much. And actually, on that note, we'll move straight into question and answers. So thanks, Dorota, for that. That was an excellent presentation, and good answering of the question that I had also. So we're going to move into the questions that people had sent beforehand. If you have any questions after this session, please put them in the chat box and I'll pick them up for the team to answer. So our first question, they're all anonymous, so you can, unfortunately on the chat box, I'll be able to see who it is, but for now they're anonymous. So question 1 is, how do we interpret a scale bar on a phylogenetic tree? I'm going to throw that to Steph.
That's an excellent question. So different tree, the scale bar could be a bit different. So like if you, may I share the screen again, maybe? Sorry, bear with me. So in a tree, in the lineage tree like this, you can see the scale bar is like 53 something. And this is indicate the scale is based on number of SNPs. And sometimes it is like 0.06 for a species tree in here. It indicates the substitution per site. And how to calculate the substitution per site? For example, in this alignment between human and mouse that I extract from the EBI website, there are 10 bases in the alignment, and there is one substitution. So the substitution per site is 1/10. So there are also trees that is measured by time, but I'm not going to further explain here. In that case, the branch is the scale is in time. So that's how you interpret the scale bar.
Thanks for that, Steph. We have another question that's just come from the chat box, and this was in relation to your presentation, Steph, so I'll let you answer this one too. How do you select a reference for a lineage-specific phylogenetic tree? I mean, it can be both you, Dorota, or anyone.
So for lineage-specific phylogenetic reference, you can choose one. Usually we will choose the genome that is referred the old more ancient. So you can either if it is from short reads, you could reorder the context to make a reference, or you could use PacBio sequencing to sequence the whole genome to make it as a reference. Yeah, I hope I answer your question. I'm happy to have a follow-up question about this.
Yeah, and I think maybe sometimes it's easier to send a specific one. Like I'm sure if it was to Dorota, you'd be like, what's a good reference for CC17 and things like that. You can answer more specifically if you know of a very good closed sequence, right?
Yeah, when we choose the lineage specific sequence for different GPSCs in Pneumo, so as I said, we choose the ancient one because it will help us to do the Bayes analysis to construct the time measured tree. And those references are all published in the recent MGen paper, and I can share the resources here so everyone can download the reference genome and do the analysis.
Cool. We're going to move on because we've got quite a few questions to cover. Again, to you, Steph, what is long branch traction? I think both of you kind of mentioned this kind of long branch, but would you be able to answer to our participant what long branch traction is?
So long branch attraction is a systematic error. So so for say distantly related isolates that that may be incorrectly they cluster together, but they are actually not related together on a tree. So when you have a long branches in the tree, you have to pay attention if it is due to long branch attraction or it is real samples that cluster together.
Cool, thank you so much for that. That was quick. We'll keep moving on swiftly. So the third question is, how do you view Fandango output together with MicroReact? So I think that's kind of a odd question, but I'll quickly share my screen because you can't really put them together. These are two fairly different programs, but they do take similar tools. So if I share my screen really quickly, and what I want you guys to pay attention to here is the header. So you put the name, which has to look exactly like the gene name you've put, and then it has to be a colon and then the color to indicate color. When you look at the data for MicroReact, and the input is again the gene name, but then you have two underscores, so representing the same as the colon. But what is interesting, you can put one you can put the micro React stuff into Fandango, but then the other way won't work. So one is more suitable for resistance data. I would kind of format it to just the micro React, because you will see, if quickly just throw in the micro React data so you can see quickly what it is. And so I'll just throw in my Newick tree, and my metadata, which is now the micro React stuff. And I'll just leave that as, so it comes out specifically like that. And if you want it to look like Fandango data, you toggle that button at the top there, and you can say show metadata blocks. So then you can just click the resistance ones you want to see, and it kind of looks like the output of the Fandango. But I hope that sort of answered the question. But yeah, ask a follow-up question if you need more help, but I'll quickly move on to the next question, which is question number 4. When we create a new project in MicroReact, is it possible to copy files from Google Docs by just sharing the link? That's a quick answer. That's a no. You have to use a CSV file. So if you're using Google Docs, you have to export it as a CSV file and then input that into MicroReact. Question 5, in MicroReact, every project you create private? And is the data published or not published? So I think that's Dorota. I'll leave. Oh, is that Dorota or me? Oh, I think that's me. I guess that's me. Yeah. Oh yeah, I'll say no. It's private for sure. It's just specific and visualized to you. If you share the link, that's what will be public to everyone. So you can share it amongst people you collaborate with, so they can see your tree. But the link is private. Question number 6. How do we construct a Newick tree? So that's to Dorota.
Yeah, so we were discussing how to answer this question, and we can have a very long answer, which would be a separate webinar, or a very short one, and we decided to go with the short one, and that we can use a number of different tools and softwares. So we're gonna use IQ-Tree in training, which is becoming very popular because it's got lots of options, and it does a lot of the hard work for the user. So it actually selects the best model for you. I mean, other tools that we've been using at Sanger, it's like RxML. There's others algorithms like FastTree, but again, this is something that we probably don't want to go into too much details of, because some of those are all command line based, and if we're not doing it in training, we probably won't be discussing it much. But there are a number of tools to reconstruct the tree, and we've picked one that is user friendly and also actually does a very good job of it.
Awesome, thank you. Oh, sorry, you could hear me tapping away. I apologize for that. I was supposed to mute it. Thanks, Dorota. That's kind of awesome. And then our 7th question: can you modify the CSV file if you're using MicroReact or Fandango? So I'll let Dorota answer that as well.
So based on my personal experience, you can't. I mean, it might be something that the MicroReact team are working on. But if you have a new spreadsheet that you've added some columns or you've revised some columns, with MicroReact you have to upload everything from the upload button again. So like as if you were creating a new project, whereas with Fandango you can actually drag and drop while your previous analysis is displayed, so it will update that. But in short answer, it's like you with MicroReact you have to create a new project.
Thanks, Dorota. And last but not least, what method, this specifically for Steph, I suppose, what method will you use for phylogenetic tree calculation in GPS?
So in the GPS, both the species-wide tree and the lineage-specific tree is calculated using a method called maximum likelihood. So, and I think there is another question is what minimum bootstrap support value was selected. So usually is 70% is a good value to support the tree topology, but sometimes the bootstrap value could be 50% could be satisfactory. I hope I answered the questions.
I hope so too. I hope whoever asked it is also fairly happy with that. A thumbs up would work also. And I think that's pretty much it for questions. And we just wanted to leave a few minutes at the end. And I'll quickly share what our next plans for the next module is, and I'll let Steve wrap up the call. So very quickly, I'm going to share my screen again. Like Dorota, my Zoom gets unsteady if I go into full screen mode. So I'll just share that. Hopefully you can see the screen. So we've just completed F1, and this is just using the drag and drop tools. Our next one, and what we're doing for F2, is we'll be talking about more pop punk, and this is what Dorota had mentioned and Steph had mentioned in their presentation, that we'll be covering pop punk and clustering, as well as talking more about why you'd use core genome, MLST, or SNP-based trees to analyze your data. There's advantages and disadvantages to either, and it completely depends on your data, right? On top of that, we'll then go into A1, which is another online module that will now be using a little bit introduction into Unix, but also using standalone tools to analyze your data. And that's where we'll be teaching you how to use IQ-tree, and then you'll be able to generate your own Newick file after that, for sure. And then the last and final will be a face-to-face course, but that will be Unix will have to be, you'll have to have had prerequisite Unix stuff. But I think by the end of A1, everyone should be able to analyze data and write up their data into a full-on publication level. So we should all be good. So I wanted to break down F2 a little bit more. So we'll be using Pathogen Watch for analysis. In F1, we just use it for visualization, but F2 will now be using for analysis. So this will be species identification, MLST, AMR and serotyping specifically for strep pneumo, as well as pop punk, but also strep pneumo specifically. But John, who's created pop punk, he's created pop punk in the program, has also created a web version, which would be drag and drop, but we can both use this for strep pneumo and strep galactii. So that's all good. And then last and not least, we'll be doing more genotyping. And this is, I guess, specifically for GBS. So you can do AMR and virulence typing standalone. Right, I think that's it from me. And I'll hand over to Steve to wrap us up and bring us home. Thanks.
Okay, so thank you, Christine, and thank you everyone for joining us today. I just wanted to say a few things in closing. First one is that Christine has done a great job in putting these training modules together, and she has really good experience in genomics training generally. But any feedback that you have would be really welcome. Feedback on how the modules are going so far, feedback on this webinar, and it can be positive or negative. We like to hear positive things, but the negative is also really useful to help us improve everything as we go along. We're really hoping that these webinars can become a networking and sort of community activity. So as well as having webinars associated with the training modules themselves, we're thinking that maybe any member of this group can give a presentation to this group. So if you have anything that you'd like to present to this group, then just let us know, and we can add those things to the agenda going forwards. Okay, so thank you all very much for joining, and we'll see you next time.